I didn't miss one before. I don't know.
Recording in progress.
I'm going to ask this one more time. Who is iPhone 2?
Michaela.
Okay, Michaela, I know you've told me that before. I just have to remember because I'm taking roll one more time.
My name on here says Stacy Ozuna, but it's supposed to say Stacy Thomas.
Oh, I've got you down. It actually is Stacy Ozuna.
Yeah.
Yep, I got you.
Okay. Thank you.
You're welcome. Bear with me. No, don't leave reading. Let's see. Who is still on their 2-week access with the lab manual?
I am.
I am.
I am.
Jen-
I
All right, tell me, okay, Genesis,
Bianca.
Bianca,
Michaela.
Michaela,
Hannah.
Hannah,
Aaron.
Hannah, and Aaron. I have a favor for you. What I want to do, and I'm going to share with you my email. Give me 1 second here. Is really quickly, give me 1 moment here. Or oh, that wasn't what I meant to do. Cancel. Bear with me just for a second here. Do it one more time. Okay. Let's see if we can go in now. So what I'm being asked to do is get a screenshot of what it looks like when it declines you access. And bear with me just for a second here. Not sure if it's gonna bring up the right email. Here. See if it brings up the right email. Of course it doesn't. No. It is asking me to send them a screenshot. I'm being asked to send them a screenshot of what it looks like when it declines you. And when it does not allow you access in. And because I don't see what you see, I think we've had one student show me. But that was the one time that we did it together. And I'm trying to describe to them that it just it's declining you access. And I've been asked to send them a screenshot saying what it looks like. And this is not gonna work. Bear with me just for a second here. Keeps wanting to pull up one school's email and not this school's email. So give me one moment. This is what I get. One second here. So bear with me. So, okay, so I'm being sent a YouTube tutorial. I've been emailed a YouTube tutorial about how to get in and get access to the thing. When it declines you, if it declines you after you follow this YouTube video, if it declines you, I want you to send me a screenshot showing it's declining you. Is that clear what I'm asking?
Mhm.
I will email that to you today. But when it declines you, I need you to send me an email say or send me an email showing that the video and what you see on your screen are not compatible. That maybe the video is asking you to do things that you don't even have access to. I want you to send me a screenshot showing me that. And we'll work it through from there. At this point, I've kind of quit just trying to email people. I call them on the phone and email them. So we'll keep trying to figure it out. But let me I don't. Yeah, send me an email saying what it looks like, and we'll go from there. Because I don't think, from what I saw before, all you should have to do is click on the, bear with me here. All you should have to do is click on the assignment. And when you click on the assignment, it takes you into the lab manual. And it doesn't ask you to go through what's called the red shelf. It doesn't ask you to do that. And so I'm not sure what they're asking you to do. So I'm going to send this to you and ask you to show me what you're seeing. Because what I think, and you can correct me if I'm wrong. So this is the assignment from the manual today. It's going to give you a series of questions. When you click on that, doesn't it take you right into the manual? And if you don't have an account, it asks you to create an account. Am I right?
Yes.
Okay. Send me a screenshot when it declines you. You have access this week. You may not have access next week. We will work things out. But I want you to send me, go ahead, I'm sorry.
Sorry. I already created my account, but it just tells me that I have 13 more days.
Okay, well, after that 13 days, when it declines you, I want you to send me a screenshot immediately. Can you do
Okay.
that?
Mm-hmm.
Okay. And we will work this out. I apologize that it's happening, and it isn't something that I could have controlled or I could have foreseen, but we will work through it. Any other questions or concerns? You will today with lab, we're gonna have a little bit of a mini PowerPoint and watch some videos. You have 2 virtual labs. If you look down here, you have 2 virtual labs. So those are on the McGraw-Hill Connect. And here's your assignment. And these are 3 videos that are going to go along with the PowerPoint today. I'm going to click on the PowerPoint here. So with today's lab, we eventually, as we keep moving through the semester, the labs are going to become so where they are. They're going to start coordinating better with the lab material, with the lecture material. Right now, we're still off a little bit, and that's the way it is all of the time and most of the time, especially at the beginning of the semester. We can't always have them be completely compatible, but as the semester goes, they will become more and more compatible. So today we are going to talk about microscopy, and we're also going to talk about cell structures, which we will spend a lot of time talking about in lecture in about 2 weeks. So today is kind of a overview summary of what's coming up in lecture. So lab exercises 3 and 4. the first part is knowing the parts of the microscope. When we're looking at a microscope, we have a head piece, we have an arm, and we have a base. Bear with me just for a second. I have a little tiny mini microscope. I'm going to actually turn, I should stop sharing just for a moment here. So I have a little tiny mini microscope. And this microscope I actually purchased at a thrift store for, I think, $5. And it's called My First Microscope, and it probably, I don't know who makes it. But on a microscope, we have a head, we have a base, we have an arm, and we have a base. When you hold a microscope, many microscopes, this one is actually, like I told you, I got it for $5. But most microscopes can range from $100 to several hundreds to thousands of dollars. So when we carry them with care so we don't break them, we don't drop them. So one hand on the arm, one hand on the base. Don't carry them like this. One hand on the arm, one hand on the base. With regards to light microscopes, which are what we're most familiar with, inside we have two different lenses. We put our sample on a slide. We put it on what's called the stage. And we have little stage clips that hold the slide in place. We have a light. I'm trying to remember if this actually, if I've got this so the light actually works. Nope, I don't. Just for a second here. Well, it's got batteries in it. Why does it not? Maybe the batteries are dead. Like I said, this is a cheap little microscope. Oh, no, it does have a light. I don't know if you see the little light, but this light, because this is a cheap little microscope, comes from the top. On more real microscopes, it comes up through the bottom.
Mhm.
So it comes up through the bottom. It shines through a really, really thin specimen. And when you look through the eyepiece or the ocular lens up here, the eyepiece that contains the ocular lens, if you think ocular meaning eye, like otic, if you remember that from your anatomical regional terms, otic, ocular piece meaning your eye. So you look through this, you look through the ocular lens that's in it. Oh, I'm wrong, oculi. Oculi is the right one. My mother's correcting me. Otic meaning ear, oculi meaning eye. Oh, I'm sorry. Give me a second to get my head on straight here. So ocular lens. And then we have an objective lenses. We go from a low power scanning objective lens. So this is the lowest magnification, to a high power lens. One of the first things about microscopes that we have to learn is how to calculate the total magnification that we're seeing. What we do is we calculate the strength of this lens, multiply the strength of this lens by the strength of this lens. This lens, when I put it on the lowest power, which is, remember, that's my low power objective lens. Objective lens, meaning the lens that's going to be on the object that I'm looking at through the slide. So this is my objective lenses. This, the lowest power objective lens, and I don't know if you can see that really well, but it says 4x on there. On my ocular lens, at the very, very top, it says 10x. So when I want to calculate the total magnification that I'm seeing when I'm looking at the slide, how high is the view being magnified? It's 10 times 4. What's 10 times 4? Someone tell me. 40. That's my total magnification. One of your questions, and I'll tell you that this will be on the lab, your first lab quest, is to be able to calculate your total magnification. If I give you the ocular lens strength and I give you the objective lens strength, what is the total magnification? And that's it's fairly easy. Now, to explain just a little bit more of how this microscopes work, because I have two magnifying glasses here. Two magnifying glasses. One of these is my ocular lens. With it alone, I can magnify a little bit, right? I can see, when I'm looking down at something, I can magnify a little bit. However, when I combine it with another lens, whatever I'm looking at becomes much bigger, because the magnification of one of the lenses is multiplied by the strength of the other one. That's the same way that it works in light microscopes. So the strength of one of the lenses is magnified and multiplied by the strength of the second lens. Is that clear?
Mm-hmm.
I mean, and if you're lucky, fortunate like me at my house, where I can find two different magnifying glasses in my apartment, you can do that same thing and see that that's the way it works. Once one magnify, like I wear glasses, and with my glasses alone, they help magnify things. But then if I put the second lens up to my eye, it magnifies it even further. The strength of my glasses are magnified by the strength of the magnifying glass, so that what I see is even bigger than it was before. With that in mind, let's come back and look at this one more time. So I have a focus knob, and on this one, it's a pretty simple focus knob. This little knob right here that moves the stage up and down. This is called my focus knob. On more expensive real microscopes, you have a coarse adjustment knob and you have a fine adjustment knob. And the fine adjustment knob is a smaller knob within this knob. And we'll see that here in a second on the video. You have light coming through the bottom.
Light.
Yep, you have light coming through the bottom that can be condensed with a condenser that's right underneath your stage. So you can condense that light onto your specimen. So you have a little hole in the bottom of your stage that shines the light into your specimen. With regards to light microscopy, your sample has to be really, 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 really thin so that the light can pass through it. When what you are seeing in the, what you are seeing when you look through the lens is a light refraction pattern off your specimen. So if you remember light waves, when they hit an object, they become refracted and they get bent. And what you're looking at is the pattern of bending of the light waves. So that's the same thing you're observing here. When you first start with a specimen, you always start at the finest objective. Start with the lowest objective, so lowest magnification. Get your object on your slide so you can see it underneath in the microscope. And then move up in higher magnifications. Any questions about what we just went through? Probably at some point when I have paid off some of my bills and making a little bit more money, I'll probably get a more expensive microscope to keep at my house because I sure do enjoy this one. So I have a head. I have a revolving nose piece that allows me to change the objective lenses. I have the aperture right here is with the hole where the light comes through from the bottom. I have the light. I have an iris diaphragm and a condenser, which work to condense the light onto my specimen. I have a coarse focus knob and I have a fine focus knob. And I can change the light intensity that's coming up through my specimen by adjusting this knob. One thing that you'll learn, and when we're allowed to all be back in class again, or you're allowed the first time you use a microscope, is you only use when you're focusing on an object using, when you first start focusing on an object, you only use the coarse focus knob on the lowest magnification of the objective lens. So only on that 4X lens will you ever use that coarse focus knob. Once you start switching to a higher strength objective lens, you will only use the fine focus knob. And that is so that you don't possibly smash your objective lens through your slide, because that coarse objective knob is going to move the stage a lot more in magnitude than your fine is. And it has the you could capably, potentially drive your objective lens through your slide. And so you only use the fine focus knob once you are at once you are no longer using you only use the coarse focus knob when you're using the lowest power or the scanning objective. So with this in mind, bear with me just for a second here. Oh, I don't. And watch a little mini video talking about the parts of the microscope.
My job is to.
Oh, bear with me. Shoot, there.
This demonstration is showing you the parts of a microscope. We have the eyepiece or ocular. This is a binocular microscope, meaning that there are two eyepieces.
So
These eyepieces can
my microscope has monocular, meaning it has one ocular lens. Binocular, meaning that it has two.
be moved in or out to accommodate the distance between your eye.
So that's one of the, when you're dealing with a binocular microscope, that's one of the first things you do is adjust it so that it's the ocular lenses are the same distance apart as your two eyes.
We have the arm of the microscope and the base. On the bottom right-hand side is the on-off switch. And on the left side of the base, you're going to find the rheostat or the dimmer switch.
So that's your light intensity knob, your rheostat or your
This
dimmer
dimmer
switch.
should be rotated so that it's closest to you. And if you needed to dim it, you can rotate it in the opposite direction. We have our revolving nose piece that consists of 4 objectives. The scanning power with a red color coded band.
Scanning power meaning the lowest objective, at the same time, the lowest strength objective, the lowest power objective.
The low power objective with a yellow color coded band. The high power objective with a blue color coded band. And then finally, the oil immersion lens with a white color coded band.
Chances are, at least in your college adventures, you will not deal very much with the oil immersion. Normally, they often will take them off microscopes in colleges so that students don't destroy them. Because like I said, we're getting into very expensive tools here.
When you're done using the microscope, the scanning power should be left in place.
So after you're done, you put away your specimen, you move your stage all the way down. Move your stage all the way down. And make sure that your lowest power, your scanning objective is in place.
Stage of the microscope, the slide clip that holds the slide. Move the slide. Underneath the stage on the right-hand side are 2 knobs that allow you to move the slide back and forth or side to side. Below the stage is the condenser, which consists of a series of lenses, as well as a diaphragm that adjusts the light as it goes through the slide. On the base, you'll find the light or the illuminator. to move the stage closer to the objectives, on the arm, you're going to find two knobs. The course adjustment knob that moves the stage in large increments and should only be used for the scanning power. Connected to the course adjustment knob would be the fine adjustment knob that moves the stage in finer increments.
Are there any questions right now about
Good grammar and spelling are important.
are there any questions right now about the parts of a microscope and their functions? Sure. Let's see here. So some term vocabulary. Monocular. Do you remember what I said monocular meant?
A single lens.
Yep, it means that we have a single lens.
Mhm.
So binocular means that we have a single ocular lens. So binocular meaning that we have two ocular lenses. There is the scanning objective lens, which is your lowest power objective lens. Then she refers to it as being a low power objective lens. So that's your second one. Your second higher magnification. Now let me say it here. So you have your lowest power magnification. You have the next increased magnification. And then you have a high power magnification. So when you're done with the microscope, you want to make sure that your stage is all the way down to the lowest part it can be. And your scanning objective lens is in place. Then you have a low, then one thing that we didn't cover, but I hinted to it, but I didn't say it. Microscopes are set up so that they're parfocal. What that means is that once you have an object in focus, with the rough adjustment knob, focus knob, and the fine focus knob at the lowest magnification, so you're scanning objective. When you switch up to a higher magnification lens, your specimen should still be mostly in focus. you should only have to alter your fine focus adjustment knob to bring it into focus again. So parfocal means that on, are you looking for Paisley? She's right here asleep. So on the lowest, so let me repeat that one more time. She came up here at the beginning of my class. On the lowest magnification, we would get our specimen into the best focus we could using the coarse focus objective knob and a fine focus objective knob. And then when we switch to a higher magnification, our specimen should still be in fairly good focus. Should still be able to see it fairly well. That's what parfocal means. Magnification is how big is the specimen, how much larger can you magnify the size of your specimen and what you see. So how much more can you enlarge what you're looking at? Resolution is being able to distinguish one part of your specimen from another part of your specimen. So for example, if I, bear with me. I take off my glasses and bring my finger about right here. All right here, my finger is in pretty good focus, maybe about right there. A little blind. So it's in fairly good focus. The closer I bring it up to my face, the more blurry it becomes. So I can see it a lot better. Like the size of it is a lot bigger than what it is when it's down here. But the edges of it become really blurred. My resolution goes down. When I can't see the edges very well and I can't see the detail because it's all blurry, that's my resolution. So magnification has to do with size. Resolution has to do with how well do I see detail of it. With microscopes, it's really great if you can make things really, really big. We like to make things really, really big so that we can see them. However, when we make them so big that, and all it does is become blurry, that isn't helping us, right? When we want to be able to see the detail of a specimen, it doesn't help us to make it really, really big and not be able to see any detail. So the strength of, and the reason that we keep making bigger and better microscopes is because we're looking at resolution. We can make things really, really big, but when it becomes all blurry and we can't see any detail, we want them. The whole reason we use microscopes is to be able to see detail, not to just make things really, really big. Is that clear what the differences are between magnification and resolution?
Yes.
Okay. And I've always, that I've tried to say with you a couple times, and I'll keep saying it again. If what I'm saying is not clear, please tell me. And let me try to think of other ways to say what I'm saying. The field of view is what you see. Bear with me just for a sec. The field of view is what you see when you're looking at your slide. And when you're looking at your slide and what you're seeing on your slides. When I increase the magnification, so I change I switch up my objectives. I only see I see a smaller section of my slide specimen, but it's bigger, it's magnified. So as I move up the strength of my magnification, my field of view goes down. Field of view meaning what I'm able to see on my slide. So as I increase the strength of my magnification, my field of view is going down. One thing that we're not really going to talk about. Well, I could. Give me a moment. Actually, let's do that. 1 second here. This video I haven't put up so that you can access it, but I will. Give me 1 moment here. So this is a link looking at a slide.
My job is to make college. All right, in this video, we are going to be looking.
So when you put a specimen on a slide and slip it in and put your stage clips over it so it doesn't move, it is inverted with regards to what you see is inverted with regards to what's there. So you put on the letter E on a slide, you slip it under, stick it down on your stage with the stage clips. And even though the letter E is right side up, underneath the microscope, it's switched. It's flipped.
Looking at the famous letter E slide, almost every class that uses microscopes will start with the letter E, just a piece of, from newsprint, wherever. I can give you the details on an activity to do this yourself. But I'm looking at, when I put the slide onto the stage, the E was right side up. It was facing the way that it would look if I was reading it. Here you see that it's upside down and backwards. So that's one of the reasons we like to use this letter E, because it shows you that as the light rays are bent or refracted through a microscope, you're actually seeing a mirror image. So just like as I move the stage of the to the right here, you're seeing the letter E.
So you move the stage to the right, and the letters moves to the left. So it's exactly inverted from what you're seeing is exactly the opposite or inverted from what you're actually doing.
Left and vice versa. So everything is backward. That's okay. I'll go into the mechanics of that, but I just wanted you to see this just as a reminder of why we use this activity. So I will go ahead and go to the next part, because the main thing I want to cover in this video is your field of view and how much smaller it gets as we move to higher and higher powers. So
this is
why
this
very
is
important to say.
sorry. This is scanning objective. This is your lowest power. So this is 4x objective lens.
Or whatever you're looking at in the middle of the field of view. So this large circle on the edge, which the entire circle I'm looking at is the field of view. Right now I'm using the scanning power, which is the 4x objective for a total magnification of 40 on this microscope. As I then jump to the low power, as you can see here, I'll get her tweaked a little bit and moved, but you can barely see the entire letter E here.
So you go up in magnification. When you go up in magnification, your field of view becomes smaller.
And that's because as I just jumped from the 4x objective with a total magnification of 40 to the 10x low power objective with a magnification of 100, my magnification went up 2.5 times. So my field of view is now 2.5 times smaller. But if we move forward, then I'm going to go to the high drive power and get to a total magnification of 400x. You're only seeing part of a letter E.
So you go from seeing the whole letter E to seeing only part of the letter E.
We'll slide a little bit here, move this around a little bit, but as you can clearly see, you're only seeing part of a letter E. And that's because as I jumped from a total magnification of 100 to a total magnification of 400, so obviously the magnification went up 4 times, and the field of view just dropped 4 times. So that's why we use the letter E to show us those two things, that the field of view is going to be greatly diminished every time you increase magnification, which is why it's so important to center whatever you're interested in looking at. And number two, the fact that what we're actually looking at here is an image that's a mirror image of what we actually put on our stage. All right, I hope that helps.
So conclusion here, as you increase magnification of your specimen, your field of view decreases. Also, what you're observing when you look at the slide through the lens is backwards to what is exactly opposite to what's on the sample slide. So if I shift the stage to the right, the specimen that I'm looking at is going to look like it's moving to the left. And I can tell you that when you're in a lab and you're looking at a microscope, that might sound kind of confusing how that would work. But after maybe an hour or even a half hour of practice with the microscope, you get used to it and you figure out how to work with it. Before I move on, give me 1 second here. I want to let me just see. Just a little bit more. And no, I didn't put that slide up. I'm obviously I'm looking at another course here, but I will make it so that you have access to that video. Let's look at one more video. Oh, let's look at this one. This is a good one. Oh, it won't work. Never mind. That's why I didn't put it on there. Okay. So coming back. To our class. And you will see, so on the content on D2L, I should say this before I keep going here. I put the lab exercises 1 and 2 in a separate module. Well, I made kind of an error last night and I thought, well, you know, maybe I'll fix it later, but then I thought it actually makes more sense to leave it the way I've done it. And so what you will see is that under a week, this week, I've made a sub-module, made a mini module that's got the lab exercises. And everything that we cover in lab will be in here. I'm gonna do it like this from here on out. So when you're looking for the material that we've covered, anything we've covered this week on a particular week, you're going to find under one module, meaning the week that we're looking at. Is that clear? Okay, I don't see any faces here.
Yeah.
Okay.
That works.
I think that's better than what I had before. So now, one of the most common ways that we view specimens most common ways that we view specimens is we use what's called a wet mount. What that means is that we remember we create a really thin specimen, either slight, in this case, what they've done is swab the inside of someone's mouth. Add a drop of water and place a cover slip over the top of it. And one thing, in addition to adding a drop of water, they add a small amount of dye. So this next video that we're going to look at just going to minimize that. describes that process that, and shows that process of what I was just describing here. So this is a slide showing how to create a wet mount slide, or a video showing how to create a wet mount slide.
In this video, you will learn how to stain eukaryotic cells. To make a wet mount of cheek cells,
If you'll see on here, he's got, so he's going to show you cheek cells, but if you look up here, he's got a piece of onion. That's often what we use to, the first time that you look at plant cells, is you take a really, really tiny thin piece. And like I said, it has to be very, 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 very thin. And recognize that when you're using microscopes, the smallest amount will go is usually best, and the smallest amount will go a long way. If it's too thick, you're not going to be able, it's not going to work for you because the light has to pass through it.
place a drop of fast blast DNA stain on a microscope slide.
So a little bit of stain.
Use the blunt end of a toothpick to scrape the inside of your cheeks. Place the toothpick into the drop of stain on the slide and swirl it to release the cells into the stain. Prepare the wet mount by lowering the cover slip over the stain, starting at a 45-degree angle. Avoid trapping bubbles under the cover slip.
You'll see what the way he put down that cover slip. I'm going to back up just for a second here. Or I'm going to try to back up just for a second here.
Flip over the stain, starting at a 45-degree.
So he's starting at an angle. When he puts down the cover slip, he starts at an angle. And the reason he does that is to minimize how many air bubbles he traps underneath the cover slip. Because if you do it at an angle, all of the bubbles will end up going out the opposite direction, hopefully, theoretically. You may have maybe a few, but you won't have as many as if you just drop this cover slip right on top.
Angle. Avoid trapping bubbles under the cover slip. To make a wet mount of onion cells, place a drop of fast blast DNA stain on a microscope slide. Take a small piece of onion.
Like I said, the smaller the pieces, the better chances you are of getting a good slide specimen.
Peel a piece off the inside epidermis of the onion leaf scale that is roughly 1/2 to 1.
Really, really, really tiny, and I'd even go tinier than what he's showing here.
Centimeter squared. Place the piece onto the drop of stain on the slide.
I can tell he's done this a couple thousand times, that he knows he's confident enough with that big of a piece of the onion. I personally would never do that, because really I can't stress this enough. The smaller the specimen, the better chances you have of getting a good slide preparation.
Add another drop of fast blast DNA stain on top of the piece of onion. Prepare the wet mount by lowering a cover slip over the stain.
So again, he's doing it at an angle.
Starting at a 45-degree angle. If a bubble forms under the slide, additional stain may be added along the side of the cover slip.
And so the result is the result after he's prepared that cheek cell. Bear with me just for a second here. Is this. Using that DNA stain. Right about right here are the boundaries of one cell. So the plasma membrane of one cell. And the nuclei right there, where the DNA is going to be contained. Now, you had two questions. Why do you think he needed to add a stain?
For it to be like more visible or whatever.
Yeah. Let me, Mom, will you grab me, would you grab me a Q-tip really quick from the bathroom? I'm not sure I have any toothpicks, so we're gonna use a Q-tip for this. You want to grab me a toothpick instead? I'm glad I have a lab assistant or a teaching assistant here. That works. Oh, well, grab me the Q-tip instead. Grab me the Q-tip instead. Oftentimes when they do a cheek swab, they use a softer thing. They don't use a toothpick. So I think I've said this, I like CSI shows. I like mystery shows. I've been dabbling a little bit with dabbling a little bit with anatomical forensics. So I'm enjoying this study of forensic sciences. So I have a toothpick here. And you can see my toothpick it's white. I do my cheek, and normally when they do a cheek swab, I don't think they use a toothpick. I think they actually use something with a softer edge because you don't want to gouge someone's mouth. My question for you. Okay, so I just swabbed the inside of my mouth. Is it got any color to it? It was white before. Is there any color on that now that I've swabbed my mouth?
No.
Okay, try again. What about now?
No.
Because the reason is because cells are clear. Unless I've got blood in my mouth, which I can tell you, I brushed my teeth before class. I didn't see any blood in there. Your cells are clear. So the only way that you're going to be able to visualize them under a microscope is with some form of stain. That's the reason we use stain. We have to add some sort of dye that's going to adhere to what we want to be able to see under the microscope, or we're not going to be able to see anything because it's going to look clear. Thank you, Mom. My online classes go a lot better when she's home, can I tell you? We won't say anything, though. So with a specimen like this, you can see the plasma membrane. You can see the nuclei. If we increase the magnification, and used maybe a different stain, there's lots of different stains out there, we could see other structures. So kind of a summary, compound light microscope. So a compound meaning we compound the strength of one lens by the strength of another lens. Think of compound as meaning a magnification. So one lens's strength is compounded by the strength of another one. So two sets of glass lenses, one right here, one right here, or one right down here, and one down here. So three sets of lenses pass through and focus light rays onto a specimen to produce the image you observe. That's the short and simple summary of what a light microscope does. It uses light and it focuses that light onto your specimen. And we work to condense that light using the condenser lens before. These two are really good. The ocular lens and the objective lens are really good at magnifying. We use the condenser lens more to increase our resolution. Because you remember what I did with my eyes here. It's really easy to make something really big. But then when it's so blurry that you can't see any detail, you've lost the resolution. So with that in mind, a transmission electron microscope, instead of using light waves, and in come lab lecture on Monday, we'll spend a lot of time talking about electrons. You're going to learn more about electrons than you ever wanted to know. Electrons move in wavelengths. They move in streams of wavelengths. And electrons have a negative charge. And so they have a charge, and we use, instead of using glass lenses, we use magnetic field condensing lenses. So we create a magnetic field. So we force the wavelengths of electrons to pass through the specimen. And just like light rays, they're diffracted off. They're diffracted off and they come down on the specimen. The electrons come down, they get focused, they get more focused, they get more focused. Let's see, where's our specimen? Okay, so they get focused here. And then they hit our specimen and they get diffracted. Those wavelengths of electrons get diffracted. And what we're seeing is a diffraction pattern of the electrons on the screen. So the major difference here between a compound light microscope and a transmission electron microscope is that one the compound light microscope uses glass lenses. And it uses light wavelengths. A transmission electron microscope uses magnetic field lenses. And it uses electron wavelengths. So you can see how they're very similar. They both work to condense wavelengths. One of them, the light microscope uses light wavelengths. Just for a second here. It uses light wavelengths. The transmission electron microscope uses wavelengths of electrons. And with the wavelengths of electrons, the detail, the resolution within the slide goes up exponentially. So we can see structures inside the cells that we can't see with a compound light microscope, with a transmission electron microscope. So if you think, shifting up here just for a second. You see, here's our slide. We see the nucleus. We see the kind of the boundaries of the plasma membrane. When we use a transmission electron microscope, here's our nucleus. Here's all the structures within the cell. So you can see that the detail that we can observe with the transmission electron microscope goes up drastically. Bear with me just for a second. I know I have a student who's trying to get into class here, and I'm trying to get them in. Oh, let's see here. Jessica, are you here?
Yes, I'm in here.
Okay, I'm so glad, because I was thinking she's waiting and I can't figure out how to admit her. I'm glad you're here. Okay. So moving forward, and let me close this out. So we got a compound light microscope uses lenses to focus the light wavelengths onto a specimen. We have a transmission electron microscope that uses electromagnetic field lenses to focus wavelengths of light, electron wavelengths. So just like using magnets, these are lenses that are magnets that can create a magnetic field that focus the wavelengths of electrons. Now, a scanning electron microscope, one thing that I'll just to compare here, transmission. What does it mean to transmit something? If I want to transmit a message to another planet, what do I do?
You're sending it.
I'm sending it and they receive it on the other end, right? hopefully they receive it on the other end. One of my favorite movies is Contact. It's a really old movie with Jodie Foster. I love that movie. But you transmit a message to another planet. And they read it and look at it and bring it back. That and send you back a message, hopefully. So transmission meaning you're passing electrons through the specimen. They're moving all the way through the specimen to the other side. A scanning electron, the electrons do not pass through the specimen. They get bounced off. So you scan a wavelength of electrons across your specimen. But in this case, your specimen is coated with a thin metal layer. And that thin metal layer, because metal and atoms contain electrons, when you shine a beam of electrons that you've condensed, just like in the case of the transmission, we have these electromagnetic condenser lenses. That wavelength, that condensed electron wavelength shines off the metal layer that you've coated the sample with. And it activates electrons in the metal that then fly off and start bouncing off and refracting off. And those electrons that are activated within the metal that you've coated this specimen with, so that the electrons can't move right through the specimen, bounce off and create the image that you see. So the major difference between this, a transmission electron microscope, is that the electrons are going to pass through the specimen in the case of the transmission electron microscope. So that you can see inside details inside the cell. The scanning electron microscope allows you to see really good surface details, but you can't see anything inside the specimen because the electrons are not going to pass through the specimen. They're going to bounce off the surface. Is that clear? So transmission, meaning you can transmit electrons through the specimen. And then they get bent. And the image that you see is the electrons bouncing off structures within the specimen, within the cells. In the case of the scanning electron microscope, the electron wavelengths that you're shining at your specimen bounce off the surface because you've coated your specimen with a metal. And when they bounce off the specimen, the pattern with which they bounce off this specimen is what you observe in your image. So just like when we look at an object, we're seeing light refraction. When we're using an electron microscope, any electron microscope, we're looking at the refractions of electron wavelengths. Any questions about this? Now we're going to shift gears And we're going to spend the rest of the time today looking specifically at cells. And the first thing I want to do is I want to go through different structures within the cell. So the first thing that we're going to talk about is the plasma membrane here. It goes around the whole membrane, goes around the whole cell. This separates the inside of the cell. This is a boundary that demarcates the inside of the cell from the outside of the cell. Inside of the cell contains cytosol and cytoplasm, which contains organelles. We talked a little bit about organelles in chapter 1, the things that come together inside a cell that allow a cell to function a specific way. So we have a nucleus. Our biggest organelle is a nucleus, which is enveloped within a nuclear envelope. And within the nucleus is chromatin. Chromatin is our DNA, our genetic structure. Another thing that we have in the nucleus is called the nucleolus, which is where ribosomes are made. And we'll talk, today we're going over very briefly the structures within a cell. In 2 weeks in lecture, we are going to spend a lot of time talking about these structures. So for today, we're going to learn general functions of these structures. And then when we hit it in lecture, you guys are going to be pros because you're going to understand exactly what we're referring to because we've talked about it today. So the next organelle, we have the nucleus, contains chromatin, the DNA genes that dictate the structures of our body and our cells. The nucleolus, which is going to create ribosomes, which are going to allow for proteins to be created from the genes. We have smooth endoplasmic reticulum, which helps to create lipids, meaning oils, and steroids. We have rough endoplasmic reticulum, which is involved in processing the proteins that are created. We have ribosomes that can either work, that are created in the nucleolus here, that can either work free in the cytoplasm, or they can be adhered to the surface of the rough endoplasmic reticulum, depending on what proteins they're making. We have a Golgi apparatus, which further processes proteins that are made in the rough endoplasmic reticulum and packages them for wherever they will be sent. Whether they're sent to be secreted outside of the cell, or whether they're made to be sent to one of the other organelles in the cell. That's what the Golgi apparatus does, is it processes things and packages them so that they get kind of like the post office, packages the mail, the proteins, and make sure that they get sent to the right place. And mitochondria, lysosomes, if you think to lyse something, have you ever heard, they've told to pop a pimple on your face, kind of nasty, but to pop a pimple on your face. If you've ever heard to lyse a pimple, L-L-Y-S-E. Lysosomes break down and destroy things. Centrioles are involved in making cytoskeletal elements like microtubules. If you think cyto as cell, cyto means cell. Skeleton, if you think about what our skeleton does, it gives us shape and form. That's what the cytoskeleton does for the cell, is it gives it shape and form and allows it to move and allows transportation of things within the cell. Let's see here. Notes. Two of the cytoskeletal elements, and we'll talk about one more in the next few slides, microtubules and intermediate filaments. So these are the fibers that make up the skeleton. Peroxisomes. If you're familiar with hydrogen peroxide, I know that some of you work in a healthcare field. I know that already. Have you ever used hydrogen peroxide to clean up blood or yucky stuff?
Yes.
Why do you use hydrogen peroxide?
It bubbles the blood out.
Say it again.
It kind of like bubbles the blood out of the fabric.
It's really good at getting blood out of fabric. Yeah, I've learned that myself. I mean, you know why women get away with more murders, right?
But it will bleach your clothes, so you got to wash it out first.
It's true. Do you know why women get away with more murders?
No.
They know how to get blood out of things. That's why. But with that in mind, what it does, hydrogen peroxide destroys, it denatures things. It oxidizes and destroys. Peroxisomes use hydrogen peroxide to destroy things. So lysosomes use digestive enzymes, proteins that catalyze digestion, the breakdown of things. Peroxisomes use hydrogen peroxide. Scanning down, plasma membrane. Only, like I said, in 2 weeks, we're going to talk a lot more about this in lecture. But on the plasma membrane, we have, this is the outer boundary of the cell, separating intracellular fluid or the cytoplasm, cytosol, from the outside of the cell. And we have transmembrane proteins. Remember, transmembrane meaning a protein that goes all the way through the membrane. We have channels that allow things to pass through. We have cytoskeletal proteins that allow to maintain shape and function of the plasma membrane and also help with moving things through the membrane or moving things out of the membrane. We have glycolipids and glycoproteins. Have you ever heard of the word glycogen?
Yes.
What is it?
Sugar.
You know? It's sugar. You're right. Good job. So sugar, so glyco, anytime that you see the word glyco, G-L-Y-C-O, that means some sort of sugar attached to something else in this case. So it has to, anytime you see the word glycol on the front of a word, you know that it's got to describe some sort of sugar. In the case of a glycolipid, the carbohydrate or the sugar is attached to a lipid or a fat, an oil, a fat. In the case of a glycoprotein, it's attached to a protein, the carbohydrate or the sugar. We also have phospholipids. If you think lipid meaning oil, phosphate, remember that word from the other day. This is a trace charged ion that's attached to a lipid. One thing that I'll talk about really quick, and then when we talk about it in lecture, it'll become even more clear. Anytime that you see a hexagon-like shaped thing on a diagram like this, anytime that you see that, actually, let's come back to that one more time. Anytime that you see this, and you can see it, you can see chains of it. You can see chains. Anytime that you see that, we are looking at a carbohydrate. That's what that is. It's a sugar. The sugars and carbohydrates are often interchangeable. In biology and biological terminology, we refer to it as carbohydrates, but that's what sugars are. So anytime that you see this on a structure, like one second here, not that. On a plasma membrane, like right here, that's a carbohydrate. It's a sugar. We also have cholesterol, which we'll talk about more later. So we have channel proteins. We have transmembrane proteins that can be attached to glycoproteins and glycolipids. We have peripheral proteins, meaning they're on, if you think about something, individuals being on the periphery of society, meaning they're on the edges. So peripheral proteins, meaning on the not going through the membrane, but being attached to the edge, to the sides. So we can look at the organelles within the cell using microscopy. Can you tell me, if you think back on the 3 different types of microscopy that we used, that we talked about, what type of microscopy was used to get this picture? See, it doesn't really have any color staining to it. What kind of microscopy was used for this?
Scanning electron?
Why do you say scanning electron, Aaron?
Because we have surface detail that we wouldn't otherwise be able to see with electrons passing through or with just a regular scanning microscope.
You are right, and I'm there. You're right. Except one thing, we're not looking at we're looking at inner detail, not so much surface detail, but we're looking at the inside, which is a transmission electron microscope. You're right. Good job. So we're being able to see organelles. I mean, if we were using a scanning electron microscope, we wouldn't see. Yeah, if we were using a scanning electron microscope, we wouldn't see all of the organelles. We would just see the surface of the cell. We'd probably get a very good, nice picture of the surface of a cell. Like we might see all the little channels sticking up and so forth, and the peripheral proteins on the outside edge, but we wouldn't see all of the inner organelles. So
I'm just seeing the whiteboard. I'm not seeing.
are you serious? I'm so sorry. Give me 1 second here.
It might just be me. I'm not sure.
I'm only seeing the whiteboard too. I was a little confused.
Oh, well.
I have the PowerPoint pulled up on the side, so that's why. Sorry.
All right, let me fix that for you. Okay. Do you see it now?
Yes.
Yes.
Okay. So what we were looking at was right here. So what I asked was what microscope was used here, and Aaron said a transmission electron microscope because you could see the inner details within the cell. Scanning would only allow you to see the surface. The reason that we know this isn't a light microscope is there's not a lot of color staining there. And that's when you know you're using a light microscope. Unless they've colored, which they can fake color electron microscope slide pictures. But in this case, you also can resolve a lot of detail about the organelles within the cell, which to see that you have to use a transmission electron microscope. So moving on a little bit, these slides right here, they are going to summarize the function of the different cell structures that we can observe within a cell. The plasma membrane, meaning the outer membrane, prevents the escape of cell contents, regulates the exchange of materials between cytoplasm and extracellular fluid, and is involved in intercellular communication. Microvilli increase the absorptive surface area. So these are little structures that can be on the top of a cell surface to increase the surface area of a cell. They also have widespread sensory roles. If we increase the surface area, we increase how much of that cell is exposed to the environment, which allows for it to have a larger sensory role for being able to sense what's going on in the environment. Cilia move substances along the surface area. So these are little hair-like structures that also can have a widespread sensory role. Flagella are seen in a lot of bacterial cells, but only one, and I'm trying to think if they're seen in any plant cells, I'm not sure. but they're seen in one type of animal cell. What do you think that cell is? These are the little flippers that go back and forth that allow something to swim. Don't think too hard about this. If you read the slide, the answer's right there. What animal cell? There's only one animal cell that has flagella. What is it?
The sperm.
It's the male sperm. It's the only thing that has a little flipper that allows it to swim. And that little flipper is the flagella. Cytoplasm is the area of the cell between the plasma membrane, meaning the outer membrane of the cell, and the nucleus. It includes the cytosol. And have we talked about what cyto means? CYTO. What does it mean, Tariq?
Is it cell?
It means
Or-
cell. It
yeah.
means cell. Good job. So cell, it means cell. So cytosol is the fluid within the cell, or the components within the cell. If you think of an organ like your stomach, or another organ that we have, your liver, or another organ that we have, your heart, they each have a particular function, right? So organelles are essentially your stomach and your liver, but they live within the cell. They each have their specific functions that allow for the whole cell to function, just like your organs do within your body. The cytosol is the fluid portion of the cytoplasm in which many of the cell's chemical reactions occur. Go over a little bit here. So your cytoskeleton, remember cyto, like Tariq said, cyto means cell. So your cytoskeleton is the fibers or the structures within the cell that allow it to have a particular form and allow it to be able to move and maintain a cell shape. So the cytoskeleton has three kinds of protein filaments that maintain the cell shape and are involved in cell movement and movement of organelles. The three different protein filaments are microfilaments, intermediate filaments, and microtubules. Microfilaments, the very top one here, support the microvilli, these little finger-like structures that give a cell more surface area. So the microfilaments support the microvilli and the plasma membrane. And they are involved in muscle contraction and other cell motility. And endocytosis, if you think cyto means cell, endo means to inside, or you can think to enter. So endocytosis is to bring something inside the cell. And we'll talk about that more in 1 second here. So microfilaments are involved in muscle contraction, cell movement, other cell movement, endocytosis, bringing things within the cell, and cell division, being able to divide cells. Intermediate filaments give shape and physical support to a cell. They anchor cells to other cells and to other extracellular material. So they anchor the cells to their external environment. They also compartmentalize cell contents. Microtubules, the third type of protein filament of a cytoskeleton, help to form cilia. Remember, these are the little tiny hair-like structures, and flagella, the little flippers on sperm, and centrioles and mitotic spindles that are involved in cell division. They also enable mobility of cell parts, form trackways that direct organelles and macromolecules to their destinations within cells. So these different filaments have sometimes overlapping roles. If you look there, you'll see that almost all of these, the microfilaments and microtubules are both involved in movement of organelles and cell parts. So they have some overlapping roles, but overall, all three of these types of filaments are involved in moving things within the cell, allowing the cell to move to being able to move itself, allowing the cell to be able to move and migrate, and allowing things to move within the cell, and allowing the cell to maintain a particular shape and structure. Let's take 8 minutes right now. So let's stop right here. And I will meet you back here at 11:30 because I think I need to walk away from my screen just for a minute. I don't know if it's like this for you guys, but my eyes start to get really kind of tired when I'm just staring at my computer screen. And yeah, it's a normal thing. You know, your monitor is constantly flashing lights at you, and it gets very hard on your eyes. So let's get up and move around for 8 minutes and come back to this. It's hard to follow sometimes. There's a reason I'm always having comments. They don't require that. As they're said, that they don't do a lot of this was You know, Paisley, you're lucky to just choose. That look, and then it makes you choose that kind of. Look at this look on her face. She's just, she knows she's lucky she's cute. Are you lucky you're cute? Yeah. So Stevenson mom does not want me to bring in unfixed animal organs. I don't know why they're worried about it. They don't realize it's just hamburger when it comes right down to it. I'll talk with Carol about it and see if she can't talk with them about it. Because I don't know why they think they're worried about it. They don't know how fresh it is. Yeah. Know how carefully you have it. See what happens. I could always sneak it in. Mary. But they don't know what will hurt them. Sounds like a good way to lose your job. I know. Very important to push the limits. Are your students listening to you being controversial? I don't think so. All right, let's come back here. So the nucleus basic cell structures, the genetic control center of the cell. It directs protein synthesis and shelters the DNA. It is the assembly site. Oh, shoot, it's not right. Switch these words around. I've got a typo here. The nucleolus is the assembly site of ribosomes. So this is where the ribosomes are created. The chromatin is long, thin strands within the nucleus, where each strand is composed of one DNA molecule and the associated proteins. Any questions about this? Because I'm not seeing any faces.
I don't have any questions.
Are we allowed to refer back to these? Do you give us the PowerPoints and like our lessons? Because sometimes like I just can't write everything down that's on the page.
You have access to this on D2L.
Thank you.
Should.
Did you
And,
ever figure out the reporting?
What?
Did you ever figure out the reporting?
Give me this weekend to get it done. I'll get them up.
No problem.
I should be able to get them up. Just give me a weekend to figure out how to do it. This material is also in the ebook for the lab manual. It's also in the textbook in chapter 3. The rough endoplasmic reticulum is the functions in protein synthesis and the manufacturing of the cell membranes. The smooth endoplasmic reticulum is involved in lipid synthesis, detoxification, and calcium storage. Ribosomes work to interpret the genetic code, which will the DNA sequence, and synthesize polypeptides, meaning proteins. The Golgi complex receives and modifies newly synthesized proteins or polypeptides, synthesizes carbohydrates, and adds carbohydrates to glycoproteins. Remember glycoproteins like I think Aaron may have said before, glyco means a sugar or a carbohydrate. So glycoprotein is a molecule that's made of a protein and a carbohydrate. Golgi complex also packages cell products into vesicles. Secretory vesicles are vesicles that allow things to be secreted outside the cell via exocytosis. So we talked about endocytosis is to bring things into a cell. Exocytosis is to take things out of a cell. Exo is to exit a cell. So lysosomes contain enzymes and protein catalysts that are involved in digestion. Autophagy, meaning to digest the cell to digest itself, which happens when the cells die. Programmed cell death, meaning when the cell is programmed to essentially commit suicide. glucose metabolism. Excuse me, glucose mobilization. I can't believe I said that. Metabolism, mobilization, glucose mobilization. Peroxisomes contain enzymes that are involved in detoxifying with hydrogen peroxide free radicals, alcohol, and other drugs, and oxidizing fatty acids. Mitochondria is involved in ATP synthesis. Centrioles form the mitotic spindle that's involved in cell division. Unpaired centrioles help to form cilia and flagella. The centrosome is the organizing center for the formation of microtubules of the cytoskeleton and the mitotic spindle.
What is ATP synthesis?
ATP synthesis is the energy production within the cell. We're in, I'm trying to think if we talked about it the other day, and I don't think we did. Adenosine triphosphate is the major energy fuel that the cell can use. And it's made within the mitochondria. Does that help, Lakeisha?
Yes, I just didn't know what the ATP stood
That's
for.
it. That's a good. I think I have it on another slide, but that's a very good right there. So that's a very good question. So some other vocabulary that we use here: passive transport. If you remember, we talked about gradients the other day, that things tend to move from a low to a high. And no, sorry, they move from a high to a low. Good grief, Mary. From a high to a low all the time. Unless you force it to go to not move that way, or to move things from a low concentration to a high concentration. Things always move from a high to a low unless we force it otherwise. That's what it means to be passive transport, is to move from a high to a low and to just let things do what they're going to do. You know, it's just going to move the way it wants to move, and it's always, and we always move from a high to a low. Active transport is when we force it to move a certain way using ATP, like Lakeisha was just asking about, adenosine triphosphate. We'll talk more about what that is in chapter 2, which we'll talk about in lecture on Tuesday. We also, enzymes require ATP to perform their catalytic, they catalyze reactions. They make them work faster, run faster. Endocytosis, meaning to bring things into the cell. Exocytosis, meaning to have things move out of the cell. Phagocytosis and pinocytosis are examples of endocytosis. Phagocytosis is to eat. Phago meaning to eat. So cytosis means a cell eating. So this is when one cell engulfs a large piece of material, or a damaged cell, or an infected cell. Pinocytosis is to pinch. So it's just very small cell drinking to bring things into the cell. So phagile meaning to bring in large amounts. Pinno meaning just a pinch. No pieces are brought inside the cell. So phagocytosis and pinocytosis are forms of endocytosis, bringing things into the cell. Phagocytosis meaning to bring really big pieces in, and pinocytosis meaning just a pinch, little tiny pieces, a little bit of drinking, not so much as inhaling things, a little bit of drinking, a little bit of sipping something versus inhaling and biting something off. DNA, deoxyribonucleic acid. What makes up our genes? Our genes are organized into long strands of chromatin. Chromatin, especially during cell division, condenses into chromosomes. When chromosomes replicate, because they're going to replicate, so the same genetic material can be passed from one cell to another cell with 100% accuracy. It has to be replicated. So the DNA has to be replicated. And so all of the chromatin will be replicated. All of the DNA that's contained within the chromatin will all be replicated. And then the chromatin will condense and compact up into chromosomes that if we have, once it's all replicated, we have two identical chromatids for each of our genetic chromosomes. I don't have a video of this, but I'm going to put up a video of it. Mitosis is what occurs during cell division, where the DNA, half of the DNA goes to one newly formed cell, and the other half of the DNA goes to the other newly formed cell. So this is a division of the nucleus and the components within the nucleus, the DNA and the ribosome material is going to be divided during mitosis. Especially the DNA, that's what we focus on. Cytokinesis, like Tarek said, cyto means cell. So kinesis means to cut. So this is a division of all the other components of the cell besides the nucleus. So this is division of the nucleus. This is division of cytokinesis, is division of all the other components. We have two types of cells. We have somatic cells that have one chromosome from the mother, one chromosome from the father, and we have 23 pair. When gametes are formed, gamete meaning your sex cells, the sperm and the egg. When gametes are formed, they are haploid, meaning they only contain one of each pair of the 23 pairs of chromosomes you have. So all of your somatic cells, these are your muscle cells, these are your skin cells, these are the cells that I swabbed off my cheek. they all contain one of each of the 23 pair. Excuse me, they contain 2 of the 23 pairs of chromosomes. Gametes only have 1 chromosome for each of my 23 pairs. So when I say that somatic cells contain 2, they're diploid. If you think di meaning 2, gametes are haploid, meaning they only contain 1 of each. Somatic cells contain 2 of each. So when gametes are formed, only half of the genetic material goes into a sperm or an egg. So let's give an example here. Quentin makes a conscious decision that he's going to make more sperm. When he makes more sperm, only half of his genetic material goes into each of his sperm.
And isn't it different material for each sperm?
It is, and that's a much more complicated process than I want to get into here. But it's why things evolve. Yeah. Let's not go that far. But you're right, Quentin. You're right. Question for you? What does Genesis mean? I think right here, we have someone in our class name that has a name like that. I think it's beginning. It means the beginning. What else does it mean, Genesis?
The first?
What else could it mean? If you think about Genesis, if you've ever seen that part in the Christian Bible, what does it say? What does it talk about?
I'm not sure.
It talks about the creation of the world and the creation of Adam and Eve. And the reason I bring this up is because anytime that you see Genesis on the end of a word, it means the formation of something, the creation of something. So gametogenesis is the formation of gametes. Well, gluconeogenesis is the formation of glucose. Myogenesis is the formation of muscle. Anytime that you see that word on the end of a word, you know that you're talking about the formation or the creation of something, the synthesis of something. If you can figure out what this word means, you know that it's a formation of this. So this is a formation of gametes. Moving forward. Mitosis, bear with me, mitosis is the division of somatic cells. Gametes are formed by what's called meiosis, M-E-I-O-S-I-S, which we're not going to touch on today. I think we have enough material here today. So when we refer to the whole cell cycle, we have one phase here, interphase, where 3 things happen. During this G1 phase, all of the cellular components are duplicated. They're all replicated, except for the chromosomes, the DNA. So let me back up just for a second. This process right here is essentially like it's the life cycle of a cell from birth to death. But in this case, it's from birth of a new cell to division of that cell. So the cellular contents are all duplicated, except for the chromosomes in this stage. In the S phase, which I like to think of as the synthesis phase, meaning to synthesize something, all of the chromosomes are duplicated. So we synthesize a second set of chromosomes from the one set. That's the way I remember the S phase, is I think we're synthesizing a second set of chromosomes here from the first set. We're duplicating all of the chromosomes. Then we have the G2 phase, where we look at all our duplicated chromosomes and make sure that there's no errors. We haven't made any we haven't done any error to our DNA that's going to result in future problems. So G1 phase, duplication of all of the organelles. S phase, duplication of the chromosomes. G2 phase, looking and making sure that all the DNA that's duplicated doesn't contain any errors. And if we need to, make some repairs. And then the DNA is divided in mitosis. Remember, this is the nuclear division of the nucleus. And then cytokinesis, meaning to cut the rest of the cell in half, duplicate, separate, divide the rest of the cell. So this is division of the nucleus. This is division of the rest of the cell. So during mitosis, we have 4 basic phases. We have a prophase, we have a metaphase, we have an anaphase, we have a telophase. Different components that are involved here. We have chromatin, chromosomes. We have the mitotic spindle, this little thing, that mitotic spindle that comes out and connects to the different chromosomes. We have the mitotic spindle is made by the centrosomes. And then the two duplicated pairs of chromosomes called chromatids are moved to opposite sides of the cell. And then the new cell is formed. 2 new cells are formed. So we have chromosomes, mitotic spindle fibers, and the centrosomes within the centrioles. So I like, this is Professor Gessel's mnemonic of how to remember the phases of mitosis. PMAT, prophase, metaphase, anaphase, and telophase. Sometimes it helps if I put an I above the, in front of this and say it map, because that means the interphase is first. So interphase, prophase, metaphase, anaphase, and telophase. But when I want to know just mitosis alone, it's PMAP, prophase, metaphase, anaphase, and telophase. During early prophase, so this is right after the G2 phase, the centrosomes have duplicated and the chromatin is condensing into chromosomes. So this is when the chromatin is compacting. Already had all the had all of the DNAs duplicated. The centrosomes that are going to produce the mitotic spindle have duplicated, and the chromatin is condensing into chromosomes. And the nuclear envelope that contains the DNA is the boundary of the nucleus is breaking down. Come here, baby. Paisley, you want to be up here? Pacers, come here. Come here. Come here. Prophase, the nucleolus. Remember, this is what contains the assembly site for the ribosomes, has disappeared in the nucleus, and the duplicated chromosomes are visible. The centrosomes begin moving apart, and the spindle is in the process of forming. The kinetochore, which is a center connection on each of the chromosomes, chromatids, attaches to a mitotic spindle during prometaphase. So right before, in between prophase and metaphase. The chromosomes become aligned at the metaphase plate. I like to think of metaphase as when the chromosomes are all meeting in the middle and lining up in the middle. So metaphase meeting in the middle. Anaphase, the duplicated chromosomes separate and go towards the opposite poles. Think of anaphase as being taking away. So they move to opposite sides of the cell. Telophase, daughter cells are forming. This is the end. So the daughter cells are forming as the nuclear envelope and nucleoli, meaning the nucleoluses, nucleoli meaning plural for nucleolus, reappear, and the chromosomes then decondense and become an indistinct chromatin. Up here, we're looking at an animal cell. Down here, we're looking at a plant. So focus on this because we're talking about human anatomy and physiology. So as general scheme, prophase, cells condense and become visible. Spindle fiber emerges from the centrosomes. Nuclear envelope breaks down. Chromosomes continue to condense in prometaphase, and the mitotic spindle attaches to the kinetochores on the chromosomes. Metaphase, the chromosomes are lining up at the metaphase plate in the middle. Anaphase, the sister chromatids, chromosomes, are being pulled towards opposite poles in the cell. Telophase, the nuclear envelopes material surrounds each set of chromosomes. Cytokinesis, a cleavage furrow is going to form, which is essentially a pinching off of the two newly formed cells. I have some extra videos on this that I've made available to you on D2L. One of them explains mitosis. One of them explains more about cell structures. Interphase, so when we're looking underneath a microscope, under a light microscope in this case, interphase, here's the nucleus. You can't see any real distinct chromosomes. Prophase, things are condensing, the chromatin is condensing into where you can see distinct chromosomes. In metaphase, the condensed chromosomes are meeting in the middle at that metaphase plate. In anaphase, they're starting to separate. That's probably not the best depiction of it, but they're starting to separate and move towards opposite sides. Telophase, now they're definitely separating, and you can see that they're really moving in opposite directions. Late.
Hi, Professor. Please,
Hi.
I have a question. I think the class is 12:30.
What?
I said, I think the class is 12:30.
I think it ends at 12:10. Toyin.
Yes, ma'am.
No, you know what? I think you're in the other class. Are you in 504? Toyin, which, are you in the class?
Yes,
Okay,
I have the class 12:30.
Yeah, this is my other class. This is my first class.
Oh, okay.
You can join in if you want.
I'm
but
fine.
you're-
Yes, I'll join.
Okay. All right. See you in a moment. All right. You can sit in, but know that this is the end of my other class. So late telophase, where now they're separating and the new nucleus is forming. So looking at these, prophase, seen and the little individual dart is started kind of dark. These cells right here, where you can't really see the dark chromosomes, this is interphase. Prophase, starting to condense. Metaphase, they're meeting in the middle there. They're lining up along that midline. Anaphase, you've seen them start to pull apart in the middle, from the middle. telophase, we're getting two separate cells being formed now. They're kind of still attached, but they're getting cut away, separated. Practice being able to look at a cell and distinguish whether it's in prophase, anaphase, metaphase, or telophase. Example here, interphase. I can't see any real condensed chromosomes. This, I'm starting to see that they're starting to condense. This would be like a prometaphase. They're starting to condense. I can see the little fibers coming out from the centrosomes on the side. Metaphase, they're meeting in the middle there. Seeing a very distinct line there, they're meeting in the middle. Anaphase, they're actually, this is more an anaphase. They're pulling apart. One side is one set of chromosomes is coming this way, other set of chromosomes is coming this way. Bob, if you don't want Abby in your room, shut your door a little better. Last telophase, now they're really separating apart. And after this is where we get reformation of the nuclei. Why do we use mitosis? Use it for embryonic development? Is it for child development? We need it when we're recovering from getting injured and for regular replacement of cells. One last part. The last part of your exercises today is referring specifically to transcription and translation. Transcription is the duplication of genetic material or the production of genetic material into RNA in order to convert it into protein. So transcription is the synthesis of DNA, of RNA into, the synthesis of RNA from DNA. Translation is the production of protein from RNA. And just so you know, I am well aware that that was a lot of material. There are some videos posted. One of them, bear with me just for a second here. Look. So one of these videos, this is one we looked at with the parts of the microscope. This is a video about that we saw about making that cheek cell wet mount slide, and the one that he did with the onion skin. This is one to talk about an overview of all the cell structures. This is an overview of mitosis. Last thing, I know we're out of time. This is the assignment you're going to submit. I got some complaints, and I understand about the last lab assignment I assigned you. This one is a lot less. It's only 50 questions. Let me know what you think of it. As always, I do want your feedback here. And then you have 2 virtual labs, examining mitosis with McGraw-Hill Connect, and microscopy, animal cells. Are there any questions?
So the chapter 1 stuff is due Monday, correct?
Yes.
Okay.
And the lab exercises, this lab exercises 3 and 4. Leo, you can't be right there. Nobody. The lab exercises 3 and 4A is due next Wednesday night at midnight.
And these 2 under the cell division and the mic, those 2 are those aren't lab quests, right? I'm just
No,
trying to
they're
see
not.
it on the syllabus.
They're just virtual labs.
Okay.
So yeah, I'm not McGraw-Hill Connect, just virtual labs.
Okay.
Why do
So
I
are
have-
those
Go ahead.
are those considered as like part of the lab worksheets then?
They're part of the virtual labs. If you look at the syllabus.
Oh, okay, I see them. Yes,
Yeah,
yes. The
okay.
virtual lab connect. Gotcha. Sorry.
Yep. No, that's all right. Other questions? Okay, have a wonderful weekend. Like I said at the very beginning, I'm going to make that video available for you from the Wiley Plus people about how to access the manual online for those of you who have not been able to access it still. And it's not your fault. Just so you know, it's something went wrong at the school and you were not able to access it. I'm going to make that available to you. If it doesn't work for you, if you can't follow the instructions of that video because it's not what you're seeing on your computer screen, send me a screenshot showing me that, and I'm going to send it to them. But we'll work through this problem. Have a wonderful weekend, okay?
Bye.
Bye.
A good one. Thank you.
You too.
Hello.
Hi.
I know I'm super late, but
Right?
I have a question.
Go
Okay,
ahead.
so the last lab that we did for the virtual tutorial, I didn't know that was an actual like assignment. I thought that it was just like a tutorial of how to do the labs.
It is, but I want you to do it because you're gonna have to do all these other labs.
Yes. So that's the I am gonna do it, but does it, because I know it was due last night. Like,
Kayla, I'll extend out the deadline for you. Just go in there. and do
okay.
it.
Thank you. Because I was like, I did not know that this was the actual
Will
assignment.
you send me an email? And
Yeah.
when you need to do it, and I'll do it tonight.
Yeah.
Okay.
Okay. Thank you.
You're welcome. Have a good weekend. All right.
You too.